Two key questions distinguish the first type of variation from the genomic sequence elements. Are they random? And are they related to their potential utility as adaptations in the struggle for existence? Random shuffling of genomic sequences and random fertilization, the first type of genetic variation, are correctly viewed as random and unrelated to their potential utility. But the second type of variation, modification, addition, or deletion of genomic sequence elements, although it has some random characteristics, is often non-random and is often related to its possible utility. One reason is that natural selection acts on both generative phenotypes and ecological phenotypes. Chapter 14. Generative phenotypes include toolkit elements that are modular in nature and that are frequently modified and/or reused in different ways during development. Developmental evolution, which uses toolkit elements, has non-random aspects. Another reason is that the new layers of complexity that can emerge depend on previous layers of complexity. New innovations create new opportunities for new kinds of emergent interactions. This has been previously discussed in the context of EEP, Chapter 2. A third reason is that when evolution metaphorically invents, by trial and error, a useful entity, it is likely to be useful in a variety of different ways, in a variety of different ecological phenotypes. For example, a general amino acid sequence that creates a specific pocket where reactants, substrates, can bind is likely to be useful, often with slight modifications, in a variety of enzymes. A sequence motif that targets newly synthesized protein chains so that they readily insert into and pass through biological membranes is likely to be useful for numerous integral membrane proteins, such as embedded cell surface receptors involved in signal transduction pathways. Another broad example, gene duplication and divergence and multi-gene families, will be discussed in Chapter 16. A fourth reason is that evolution will favor robustness, flexibility, and evolvability, and that means that genomic sequence elements which contribute in that way do have relationships to their possible utility. A fifth reason is that in general, exaptation using innovations for diverse ecological phenotypes that are all useful in their own contexts, and co-option are frequently observed. A sixth reason is that certain mechanisms that generate genomic variation, such as repetitive sequences which can expand, diversify, and increase the frequency of recombination events, are often hotspots for genomic changes and introduce a non-random aspect into the variation events that may subsequently occur. To summarize, the second type of variation, modification, addition, or deletion of genomic sequence elements, whether they determine protein sequences or help regulate their expression, are a type of variation that has non-random aspects that can relate to its potential utility. This should be seen as a major departure from the earlier concept of random variation in the modern synthesis. This is an important change in the UES. Genomic sequence elements do play various roles in the creative aspect of natural selection. They also help explain how a blind, undirected, and natural process can have the extraordinary creative organizing power that it does. In other words, the UES is more plausible than ever before. Beyond housekeeping genes. Apart from the housekeeping genes that keep all cells alive, most evolutionary adaptations of multicellular plants and animals depend on the expression of more specialized genes that control cellular and molecular interactions during development. This adds a major dimension to evolutionary biology that goes beyond classical Darwinian natural selection, beyond Mendelian genetics, and beyond molecular genetics. The revolutionary fusion of developmental biology with evolutionary biology is called evolutionary developmental biology, EVO-DEVO, a concept that was discussed to some degree in previous chapters.